We're going to go straight into our next talk, our final talk of this session before questions. And let me just see where we're at. So this is Rajeev Venkayya, who's president of the Global Vaccine Business Unit for Takeda Pharmaceuticals. Rajeev, over to you.
Great, thank you, Peter. Can you hear me okay?
Yes, perfect, and I can see your slides.
Thank you, and thanks for having me here today. I appreciate the other panelists' talks. They're, I believe, quite complementary to what I'm going to try to tackle, particularly Jono's at the beginning when he talked about the pandemic that occurs in 2035. You know, we are at an extraordinary time in vaccinology. Over decades of development and innovation, we've never seen the kind of revolution that we've witnessed in the past year. And I think it's worth, I'm gonna try to advance my slides here. I think it's worth reflecting on the range of lessons that we've learned. And I'm just gonna briefly touch on these big categories and give you a couple of takeaways that are most relevant to today's talk. First of all, our eyes have been opened, many of us. Peter, I know you've been well aware of the threat, and many of us have been generally aware of the threat, but clearly the world has been placed on notice that we don't just need to worry about flu as the cause of the next pandemic, but coronaviruses, I would say, have matched flu in terms of the threat. We also, as a global community, more broadly have an understanding of the animal population as reservoirs for future pandemic threats. And of course, we now have seen the power of rapid sequence sharing, and we're being reminded of that as we see these new variants emerge. We also have demonstrated the ability of new platforms to respond. Now, everybody's thinking and talking about the mRNA platforms, and I would say that for me, hands down, that is the most exciting technological development. But we also need to recognize that other platforms have come along quite quickly, and in fact have only been a few months behind the mRNA platform. Now, frankly, I think we got a bit lucky. If you think about the risk associated with platforms that require you to grow viruses in cell culture and have assays that allow you to accurately tell the quality of the product, the consistency of the product, we probably saw a shorter gap between the performance of mRNA and other platforms than we might in the next pandemic, or if we were to be tested again. But nevertheless, we have a range of platforms that have shown that they're suitable for the pandemic threat. We also have discovered a number of new strategies that we can use to speed the time to availability of product for large populations. The most important innovations here have been to take risk-based approaches to development, to do things at risk that we otherwise would do in sequence. And I would say the concept of going to where the epidemic is happening with warp speed and the way that they have established clinical trial sites across the US so that they would be prepared to go where the epi is to rapidly demonstrate proof of efficacy in a community has been really very, very innovative. Now, we've also had a number of challenges. We've seen and continue to see, particularly for the non-mRNA vaccines, that the biology of vaccine production carries risks. And inevitably, we find that there are problems in virus growth, cell growth, problems in consistency of the product, in-process testing deviations, quality control deviations that will delay the arrival of supply. And not surprisingly, with many vaccines, we've seen that there have been reductions in the volumes that the manufacturers had expected to be available at certain periods of time. We've also been reminded that assays are very hard. The tests that we use, not just on the product, but also the clinical assays to evaluate the efficacy, or I should say immunogenicity of a vaccine in humans, carry their own challenges to validate. And I'll come back to this issue of the complexity of the biology of traditional vaccine manufacturing and the uncertainty that comes with growing viruses and cells. And that, of course, is an area where the mRNA platform has a real advantage. So these are all lessons that we've learned. And in my view, they give us an opportunity to rethink how we're going to approach preparedness for the next pandemic, with the goal of the title of this talk, which is to take pandemic threats off the table. Now, one way that is being actively evaluated by CEPI is this idea of having prototype pathogens that represent, or one or more, that represent the characteristics of families of viruses that could emerge as human pathogens. There are about 23 families. We can say that flu and coronaviruses are perhaps at the top of the list, but we could go beyond that to, in advance of the next pandemic, identify a range of tools and even candidate vaccines that allow us to more quickly develop candidate vaccines to take into human clinical trials and refine those candidates according to the specific virus that has emerged as a pandemic threat. This is a schematic that is taken from a Nature article that Barney Graham and Nancy Sullivan published in 2018. Now, the concepts here, again, are not going to be surprising to Peter because he's talked about this and written about this. But I think that what Barney and Nancy did that was really impressive 2 years before this pandemic virus emerged is they laid out the exact approach that was ultimately used to develop the first SARS-CoV-2 vaccine candidate. It begins with, it basically centers on rational vaccine design, beginning with understanding the epitopes that are most important on the surface of the virus, surface of the virus and for cell entry, converting that knowledge to an understanding of what antigens or immunogens would be most effective in generating a useful neutralizing antibody response to that pathogen, applying the different platforms that we have now proven can work against, in this case, SARS-CoV-2, and then testing them in animal models in advance of the emergence of an epidemic to gain an understanding of immune correlates of protection ideally, and potentially going further and evaluating vaccine candidates in phase 1 clinical trials to assess both initial safety as well as immunogenicity and what it takes to generate an immune response, cell and antibody mediated immune response that appears to correlate with a protective response in animals. If we are able to do this, you can envision having a toolkit of vaccine candidates across multiple families, potentially multiple candidate vaccines within a given family, say the coronavirus family, that will potentially substantially accelerate the time to clinical trial material for a vaccine against the emerging pandemic threat. Now, that's not all you need to do to accelerate these timelines. There are, advance my slides, a range of other things on the vaccine development side that we can also do in parallel. I mentioned already animal models, which take time to develop and validate. That is work that can be done now. Toxicity studies on the platform itself. So if you think about the mRNA platform, actually there have been tox studies for multiple mRNA vaccine candidates that have gone into humans over the past decade. And so we had a pretty good understanding of tox before SARS-CoV-2 came along. We could go deeper, do reproductive reprotox and other assessments to further give us confidence in the safety of these candidates before they're put into humans in a phase 1 trial. Clinical assessments of safety and immunogenicity, as I mentioned, you could envision phase 1 trials for selected candidates happening before a pandemic begins. And then, of course, clinical trials that are easy to stand up quickly once you have a pandemic occurring. We can't predict where this is going to happen, but if we have the WHO, CEPI, other groups that are ready to go with clinical trial protocols in an outbreak setting, it gives us a better chance of collecting useful data with the vaccine candidate within a matter of weeks. The side of this that people don't talk much about because it's so technical and there isn't much broad understanding of the complexity of vaccine manufacturing is in the CMC space. And here the real challenge is going from what works at a laboratory scale to what you can call commercial scale. So manufacturing vaccine in the range of hundreds of liters or even thousands of liters at a time, if we're talking about traditional inactivated, vectored, recombinant vaccines. The math is different for mRNA vaccines, but here again, mRNA has an advantage because you don't have to deal with the vagaries, the complexities that go with scaling up viral vaccine, or say virus production in a bioreactor. You don't have those same complexities when you're using a chemistry-based approach like mRNA. So for both mRNA and non-mRNA platforms, figuring out and optimizing your scale-up strategy before you're presented with a threat can be very valuable. And it may not reduce your time to get into the clinic, but once you have actually demonstrated evidence that you've got a product that works, it can shorten the time between the availability of that data and substantial supply for the world. I have mentioned assays in passing. Product assays, again, are very complicated. We often call these potency assays, but there are a range of other quality control assays that are used throughout the production process that have to be validated and maintained from a quality standpoint so that you can have confidence in the process. Remember that with vaccines, the process is very much the product because we don't have good ways of characterizing the complex biologic that is a traditional vaccine. Again, this is different for mRNA. For traditional vaccines, because we can't characterize a product very well, we have to ensure that the process is robust and reproducible and that we have the appropriate quality control testing throughout the process to ensure that consistency. The final point I'll make is around manufacturing at scale. We've learned a lot about what it takes to bring new manufacturing facilities online to produce products that may not have ever been, the types of products that may not have ever been produced in that facility. It's complicated. It's complicated both because of the infrastructure needs, but also the reusable component needs, which usually vary from company to company, product to product. And again, if you're going to change one of those elements of the manufacturing process, you need to have a way to bridge to show that you have not changed the attributes of the product. But the other side of this is this concept of ensuring when the next pandemic happens that we have more distributed manufacturing capability and capacity so that we don't have the situation that frankly we're seeing today where there is an inequitable distribution of the first doses of safe and effective vaccine. We're seeing some regions of the world get early access to the vaccine in substantial quantities, while the vast majority of the world has little to no access. And that has appropriately led to many people to once again think about what's it going to take to establish self-sufficiency in vaccine manufacturing in countries in all regions, not just in Europe and the US and in parts of Asia. And here I think there is an opportunity once again with mRNA vaccines. I've talked about the complexity of traditional vaccine manufacturing, and for that reason, you can't stand up a new vaccine manufacturing capability in a country that hasn't done it before overnight. It takes time to develop the talent, the capacity, the regulatory experience and capacity beyond the time that it takes to build a factory. Building the factory is quote unquote the least of the challenges. It's these other surrounding ecosystem elements that are so important if you want to be able to maintain high quality manufacturing in a country. mRNA vaccines, because they're chemistry based, provide greater predictability, less complexity when it comes to vaccine manufacturing, and could be a gateway technology for countries that have never manufactured vaccines before to get into a form of vaccine manufacturing and essentially be a bridge to taking to the more traditional approaches to vaccine manufacturing. At least that's conceptually what to me seems attractive. You can consider it a gateway. You can also consider it a way to leapfrog more traditional ways of manufacturing vaccines. But as I say that, I want to be very clear that I am not suggesting that mRNA vaccines are the solution to all of our problems. We know that as of today, with the technology as it is, mRNA vaccines don't work for everything. We hope that that can improve over time, but certainly they've worked against this pandemic threat, and at a minimum, we will need to have an mRNA vaccine strategy to prepare for the next pandemic. And so with that, I want to bring us to a concept that CEPI is now seriously considering, and you may have seen that the UK, as the holding the presidency of the upcoming G7, has come out behind this, and that is the idea that we shrink the timeline between sequence identification and availability of phase 3 data, or let's say supply, from the 300 days incredible achievement that we had in this pandemic down to 100 days. This is the ambition. It's an aspiration. Just like landing on the moon, we don't know exactly how we're going to get there, but we have some ideas, which I've referenced, that some areas where we can look at doing things differently that could contribute to that goal of shrinking the timeline down to 100 days. It's not going to be possible for every pathogen, no matter what we do. We still don't have an HIV vaccine. So we know that there will be some pathogens that are very, very hard, but for those that are more straightforward, this is absolutely within reach. CEPI believes, and I believe, and that's going to be the North Star, if you will, for the organization post-COVID. So with that, I'll stop and happy to take questions with the group.
Well, thanks very much, Rajeev. Absolutely visionary and really exciting. And when you combine everything that's been said this morning, we end up with a vision of a future that is different and much more rosy than we've seen during COVID, which is good news for everyone on this spring day.